Have you ever wondered where the spots on dogs come from? Well, these spots actually come from a mutation in a dog's genes. The Piebald mutation is a common mutation you could see on a trip to the zoo, because many different animals, including horses, rats, cattle, sheep, and dogs are affected by it. But what is it? How can it be created? And how does it affect animals who have the mutation? Let's learn more about the Piebald mutation. The origin of the word piebald is a combination of the words pie and bold. Pie comes from the magpie bird, which has spots on its feathers. The word bold comes from white patch or spot. These words combined make it easy to understand that piebald is a condition where an organism has white patches of skin, fur, or feathers.
Piebaldism is often defined as a rare autosomal dominant disorder in which an organism is born with an absence of melanocytes in affected areas of skin or hair due to mutations on the c-KIT gene. That's a lot of complicated information in one sentence, so let's simplify it a little. An autosomal dominant disorder is an abnormal condition that is most often passed down from the parent to the offspring of a species. The disorder of piebaldism comes from at least one parent and is passed down to the child, who is also born with the condition. A dominant trait is one that is most often inherited by the offspring.
An organism that has inherited piebaldism can get it from a parent carrying a genetic predisposition, or the gene for piebaldism. The chromosomes carry genes which code for the different parts of a greyhound's body. Piebald is a result of a copy of the gene on a chromosome with a mutation passed from parent to offspring. The c-KIT gene is on the 13th chromosome and is passed on to the offspring a majority of the time as a dominant trait. Most times, piebaldism is not caused by an environmental factor because it is a congenital disorder. This means that an organism is born with it, but it can also occur from a mutation on a gene.
Piebaldism is an entirely cosmetic disorder, as it does not affect the internal workings of an organism. In certain dog breeds, piebaldism is very common. Collies, Great Danes, Shetland sheepdogs, boxers, and bull terriers often have this mutation. Another dog breed with piebald are Italian greyhounds. Many of these greyhounds appear to have white spots of fur instead of a full brown or red coat, and this is often a result of piebald disorder. The pigment on a dog's coat comes from melanocytes in its skin, eyes, and hair. Melanocytes produce melanin, determining the color of a dog's skin and hair. The absence of melanin in certain areas of the skin will give it a white appearance. Deep inside the nucleus of a melanocyte lies the c-KIT gene, located on the 13th chromosome. The c-KIT gene is responsible for coding for a protein which regulates the equal distribution of melanocyte cells across the skin and hair of dogs.
The protein is called the KIT receptor tyrosine, but we will call it the c-KIT protein for short. A non-harmful defect of a mutated c-KIT gene is piebaldism. Piebaldism occurs when the mutation on the c-KIT gene causes the c-KIT protein to be absent. In the absence of the c-KIT protein, melanocyte cells are unregulated and randomly spread across the coat of a dog's fur, causing white spots that you can see on Italian greyhounds. Piebaldism occurs due to changes in the DNA or genetic code, particularly in proteins like c-KIT. Inside cells, specifically the nucleus, DNA contains the blueprint for an organism's development. DNA consists of 4 nitrogenous bases, adenine, thymine, guanine, and cytosine, which pair up to form the DNA double helix. Adenine and thymine bond together, while guanine and cytosine bond. The order of nitrogenous bases c-KIT gene holds the instructions for making the c-KIT protein, crucial for various functions in an Italian greyhound's body.
However, the c-KIT gene cannot leave the nucleus of the cell to create the c-KIT protein, so a process called transcription is needed. Transcription is like making a copy, specifically a copy of DNA. To copy itself, DNA unwinds, exposing its sequence of nitrogenous bases, and then an enzyme called RNA polymerase creates a messenger RNA, or mRNA strand for short, by matching the complementary nitrogenous bases on itself. mRNA is an RNA strand which copies the sequence of bases to act as a messenger for DNA so a protein can be created. The code is copied onto an RNA strand because RNA can carry DNA's code on its single strand and can leave the nucleus to deliver the message DNA has to send. Once the sequence of bases is shown, the mRNA strand aligns with the DNA to copy the code on its own strand. Unlike DNA, RNA contains uracil instead of thymine. For instance, where DNA has thymine, RNA would have uracil. The RNA polymerase matches the complementary bases on the mRNA strand, but when adenine is read, instead of adding thymine, uracil is added because RNA does not have thymine. Let's go through it together. When the RNA polymerase reads thymine, an adenine base is added. When adenine is read, a uracil base is added. And when guanine is added, cytosine is added. This process continues until the RNA polymerase copies a sequence of DNA called the terminator. The terminator stops this copying of the RNA polymerase onto the mRNA. Once the terminator is read, the DNA then winds back into its original shape with the help of RNA polymerase, signifying the end of transcription. The mRNA then leaves the nucleus so a protein can be created.
Translation is the next step of creating proteins. Translation is the process where mRNA is converted into amino acids, forming proteins through codons. Translation occurs in the ribosomes, where proteins are created. A codon is a group of 3 nitrogenous bases. mRNA conveys genetic instructions to cells using the codons present on the strand. They specify amino acid combinations, which are the building blocks of proteins. tRNA uses anticodons to create proteins. Anticodons are complementary to the codons on the mRNA strand, and they have an amino acid attached to them depending on the order of the specific nitrogenous bases found on the mRNA. The anticodons pair with the mRNA codons, creating a specific sequence of amino acids. Peptide bonds, which link amino acids together, bonds the amino acids together to create a protein. This reading process begins with the start codon, which is usually AUG, and continues until a stop codon like UGA is reached. rRNA ensures the correct order of amino acids, akin to a restaurant order taking role. In restaurants, tRNA seats customers by ensuring matching nucleotide pairs, while rRNA takes orders to verify RNA reading, and mRNA makes proteins translating base pairs. Afterward, rRNA ensures amino acid accuracy, resembling a final check to ensure everyone received the correct meal, or in this case, protein. The unique amino acid sequence affects the shape of the protein, and depending on the shape of the protein, different processes take place. The shape of the c-KIT protein leads to the distribution of melanocytes.
So how do these mutations occur in dogs? Well, the alteration in the c-KIT gene's DNA code results in the absence of the c-KIT protein. In the case of piebaldism, a point mutation changes the order of one or a few nitrogenous bases on the c-KIT gene. Point mutations occur due to mistakes during the process in which DNA is replicated. These can be the result of environmental factors or problems with enzymes involved in the process. Because of the point mutation, a nonsense mutation occurs where a stop codon is added to the strand of RNA prematurely and everything occurring after it is not accounted for. A stop codon stops the process of translation early, causing the c-KIT protein to not be created. This usually happens very early in a greyhound's life, as piebaldism is present from birth.
Because piebaldism is an entirely cosmetic disorder, the negative effects of daily life on an organism with piebald are limited. There are some, though. Using an example of humans, you may notice that humans with lighter skin, and therefore less melanin, are more likely to get sunburnt. This is true with other animals with light skin as well. While humans can protect themselves with sunscreen, animals that are not as effective at protecting their skin from harmful UV rays are more likely to cause more defects in their cells that can lead to cancer. There can also be survival disadvantages for a wild animal with piebald. Many times, creatures with lighter skin are easier to spot and hunt in the wild. This can make it more difficult for an animal living with piebald, such as a deer, to disguise itself from predators. In some cases, and particularly in dogs with excessive white markings and blue eyes, there is also an increased risk of congenital deafness that can be associated with the disorder.
Dogs living house-kept, also known as domestic dogs, can live a comfortable life with piebald if the owner makes sure to take care of them well. Many dog breeders breed dogs to have specific fur patterns, and piebaldism is one of them. Though piebaldism can have some negative effects, it can be seen as a beautiful addition to a dog's coat.